Welcome to an introduction to Omics Logic. A little bit about me. My name is Laura Murray. As Elia mentioned, I am the Educational Program Coordinator here at Pine Biotech. I have a background in gaining my bachelor's degree in microbiology, and I'm currently finishing up my master's degree in biology and bioinformatics. My focus and areas of expertise are in metagenomics, transcriptomics, and genomics. And my most recent research was in analyzing the metagenomics of deep sea hydrothermal vents and which bacterial communities reside there. So in this presentation, I will explain the current challenges and limitations in bioinformatics. I will describe how we aim to address these challenges with the OmicsLogic portal. I will describe Bloom's taxonomy for learning progression and how it can be used in bioinformatics coursework. We will explore the OmicsLogic portal and how it utilizes Bloom's taxonomy in structuring its courses, pipelines, and projects. And finally, I will describe several outcomes of the OmicsLogic portal. So what are some of the challenges that the field of bioinformatics is currently facing? To begin, this field is constantly growing and developing new tech and software to improve processing time and power. The datasets that are analyzed are also usually very large and take immense amounts of computing power and memory that many personal computers just don't have. Also, despite being publicly available, data are also usually complex to access, and coding knowledge is usually required to do so. Similarly, analysis pipelines and programs require coding knowledge that many biologists don't have. In a university setting, students have the opportunity to learn the background information of designing and implementing a bioinformatics project, but don't receive practical hands-on experience with the full workflow due to the limitations mentioned previously. Similarly, faculty faces challenges with being able to cover a diverse range of topics, especially if they are specialized in a certain area of bioinformatics, or if they are unfamiliar with bioinformatics in general. For example, a professor whose research focuses on oncogene expression might not know much about the metagenomics of seawater. So in order to overcome these challenges, the OmicsLogic portal was developed. This program aims to standardize, organize, and systemize bioinformatics training in order to make these analyses usable for any biologist or clinician. There are some traditional problems with the standardization of bioinformatics coursework. Materials for courses are usually developed in-house, taught sporadically, and are rarely updated. The OmicsLogic portal introduces new material that keeps up with new technology. These courses also need to be organized. Because the challenge is to combine several different topics together, the flow and transitions between subjects needs to be developed. This is addressed at OmicsLogic through introductory material being woven throughout the course. The last challenge we face is systemization. How can you know that someone is stuck and needs help, or allow someone to run forward? To address this, OmicsLogic uses standardized modules and interactive sessions, beginning with mastering terminology and concepts to implementing these concepts with real data sets in a project-based approach. So here's an overview of the learning process. So in order to create a clear path of skill development, OmicsLogic employs the Bloom's taxonomy levels of learning. This approach starts with the basics. Students recall facts and terminology and are able to repeat and memorize the material. With this base, the learning builds to conceptual knowledge, being able to understand concepts and apply information to new situations. The student will then be able to analyze data by drawing connections and evaluate when to use certain methods. This leads them to the highest level of learning, the ability to design and create original work. So how do OmicsLogic resources help advance bioinformatics teaching? OmicsLogic is a portal for practical and theoretical learning of bioinformatics. Composed of courses designed to take you through the theoretical knowledge, OmicsLogic takes a combination of training modules, data analysis tools, curated project datasets, and interactive sessions with mentors to give a student a clear path up the Bloom's taxonomy pyramid. No matter what the user's initial experience with bioinformatics is, OmicsLogic is a useful tool for gaining a baseline knowledge of the theory behind bioinformatics, analysis of large data sets, an introduction to basic coding languages like R and Python, or beginning a data analysis project. So these 6 courses were taken from the core OmicsLogic curriculum available online. Beginning with an introduction to the applications of bioinformatics and an overview of data types, the student is able to learn terminology and factual knowledge to build upon. From there, genomics, transcriptomics, metagenomics, epigenomics, and data science are introduced. Each course starts with important terms and concepts and then introduces case studies and pipelines related to each one. This allows the student to gain the conceptual knowledge in order to begin their own projects. So OmicsLogic also expands upon its basic core curriculum with specialization track projects. Bioinformatics methods are best learned in the context of real case studies prepared by our network of mentors and collaborators that illustrate how various bioinformatics methods and omics data types are used in current research, both in medicine and in biotechnology, agriculture, and even astrobiology. The projects are prepared by taking publicly available datasets and using them to explore and understand both the research challenge and the analysis path to extract meaningful insights from complex omics data. The OmicsLogic portal gives students options for analyzing a given dataset, either a code-dependent path or a code-independent path. These datasets and associated publications can be used in several ways. So like I mentioned, you can learn bioinformatics without coding at any coding experience at all. For that, in each project, we explain the analysis pipelines on the user-friendly Bioinfo platform for big data analysis. You can also learn the scripts behind many of the steps, learning to wrangle, clean, and visualize and analyze data in popular languages like R and Python in order to create your own analyses. Finally, you can hear about the projects and their research applications in our mentor-guided programs where experts share their experience and support research projects developed by you. So here's an example of the code-independent pipeline. The pipeline builder interface on the Bioinfo platform is a color-coded pipeline that helps establish a logical connection between the technical names of data analysis methods and the logical steps needed to build the full pipeline. Data is put in as an input file, and then analyses can be chosen based off of the type of research. Each analysis is separated into 3 colors: blue for pre-processing, red for mapping, and green for quantification. Each time you click a button, a pop-up will contain additional information useful for building the pipeline. The ease of the color-coded buttons allows programs to be run without any coding knowledge. Interactive outputs allow for easy visualization of the data. Several demo pipelines with real data sets are available for students to practice the workflow, including demos on genomic variant calling of cancer cell lines, 16S amplicon analysis on diet and its effects on the microbiome, and a pipeline for examining a phylogenetic analysis of Ebola virus. So after using the Bioinfo platform to visualize the pipelines and practice analyzing, students are then able to take their theoretical knowledge, the remember and understand level of Bloom's, and apply that in a project, the apply, analyze, and evaluate level of Bloom's. Then students are able to check their comprehension with levels and badges, certificates of completion, and tests. Similarly, faculty should be able to assign the OmicsLogic portal to their students with the understanding that it will give them holistic and comprehensive knowledge of bioinformatics, beginning with the Bytes and Molecules course and on to designing their own question-based project. So OmicsLogic employs a hybrid approach, combining learning and research. To ensure that students are building off of the first level of Bloom's taxonomy, OmicsLogic tracks progress with activities like pipelines, coding, and literature reviews, quizzes on topics, key terms, and important questions, and covers a broad range of topics like biomedical research, biotechnology, and agriculture. Then, students are able to continue their knowledge base with a research assessment, making sure they are able to understand methods, challenges, and results, as well as being able to comprehend primary research literature and recreate the methodology. They should be able to ask questions based on data exploration. This leads to the ability to encounter errors in their analysis, apply theoretical knowledge of why those errors are occurring, and find a solution. Bioinformatics uniquely combines life science terms with data science skills, which allows researchers to answer life science questions with reproducible data analysis methods. So OmicsLogic research fellows are able to ask research questions about any bioinformatics topic, leading to a diverse range of projects. From analyzing affected biological pathways in multiple sclerosis to brain metastases due to small cell lung cancer, these students are able to apply what they've learned in the coursework, work with a group of professional mentors, and submit their projects on the portal for review. So students should then be able to reach the highest level of Bloom's taxonomy, create. This is achieved through the creation of their own analysis project. For example, Semih Dolaniye, an undergraduate researcher at Bahçeşehir University in Istanbul, Turkey, who finished her OmicsLogic training, was able to work on a project involving chronic lymphocytic leukemia, a type of blood cancer that has a very heterogeneous biological background and diverse treatment strategies. From her work with OmicsLogic, Semih's work was able to be published with the Columbia Undergraduate Science Journal. Here, at first, she elucidated 870 significantly differentially expressed protein-coding genes that were involved in the biogenesis and processing of RNA. Consequently, these findings led our study to investigate non-coding RNA. 33 long non-coding RNAs were found to be significantly differentially expressed among two conditions based on differential gene expression analysis. So not only does OmicsLogic collaborate with undergraduate students, as seen with Semih, but projects with graduate students and other researchers are common as well. Here is a poster presentation for a project done in collaboration between Noguchi Memorial Institute for Medical Research and Pine Biotech. Here's our team at Pine Biotech who are involved with the project: Dr. Mohit Mazumdar, Dr. Harpreet Kaur, and Elia Brodsky, as well as our collaborators at the University of Ghana, Dr. Anita Gansa and Dr. Benedicta Mensah. This poster was on Plasmodium falciparum, the parasite causing malaria, and its apical membrane antigen, the rhoptry neck tube protein binding, which is a critical interaction for Plasmodium falciparum merozoa invasion of the human host red blood cells. Several proposed malaria vaccine candidates are designed to prevent such binding and limit merozoa entry, which have been studied with variable success at in vitro and early clinical trial stages. By performing haplotype network analysis of genomic sequencing data from sites with variable infection rates, comparing our data with malaria gen repository from another region in Ghana, and mapping genomic variants onto the protein structures, they arrived at preliminary conclusions about local variation and immunogenic epitope affecting variants for consideration. In this poster, preliminary findings were shared that suggest that genomic data can be used to develop a local strategy for efficacious vaccine design against P. falciparum blood stage infection in Ghana and surrounding regions. So OmicsLogic is proud to collaborate with many diverse programs across the globe. We also work with over 15,000 students at community college like Glendale Community College, top-tier universities like Georgetown University, as well as 20 universities around the world. OmicsLogic is also proud to partner with NAPI, the National Association of Idea Principal Investigators. NAPI seeks to elevate bioinformatics research in areas of the US that are traditionally underfunded, for example, in Puerto Rico, Louisiana, and West Virginia. NAPI puts an emphasis on inclusion, making sure that as many people have the opportunity to get involved with bioinformatics research as possible. OmicsLogic is proud to mirror that emphasis of inclusion, with adding many free course options, giving students asynchronous resources to complete whenever their busy schedule allows, and pairing them with professional mentors who can offer guidance in getting involved in the field of bioinformatics. So in this presentation, I explained a bit about the current challenges and limitations in bioinformatics. I described how we aim to address these challenges with the OmicsLogic platform. We discussed Bloom's taxonomy for learning progression and how it can be used in bioinformatics coursework. We also explored the OmicsLogic program fundamentals and how it utilizes Bloom's taxonomy in structuring its courses, pipelines, and projects. And we finally described several outcomes of the OmicsLogic program. So if you're wanting to know how to get started, the OmicsLogic portal is easy and free for anyone to register, with many courses available at no cost. There are also several upcoming programs that, as a student, you can get involved in. This is a program for data science for biomedical data. There's also one for genomic data analysis, and one on an introduction to big data bioinformatics. As a member of faculty, Please reach out to us about learning more on integrating OmicsLogic courses into your own bioinformatics curriculum. So thank you all. Are there any questions?